The overall goal of this process is to culture large numbers of cells under controlled nutrient conditions, using conventional equipment found in a cell culture laboratory. The associated algorithms allow the control and manipulation of nutrients within experimental ranges.
This method can help answer key questions for large-scale cell culture applications, such as human cellular therapies. The main advantage of this technique is that it facilitates culture of cells in large scale with uniform distribution of nutrients and gases. This allows higher cell densities than static or bulk-fed cell cultures, increases yield, and reduces cell loss. This can be done with equipment commonly found in most cell culture laboratories.
Visual demonstration of this method may be helpful to new investigators, as these techniques used can be difficult to master due to the large number of steps that must be performed in a specific order under sterile conditions.
Collect the 5 primary components needed for the feeding system: a medium reservoir, peristaltic pump, stirred bioreactor, waste reservoir, and custom-designed tubing sampling set. Establish the medium reservoir using a 1-liter glass bottle, the waste reservoir using a 2-liter glass bottle, and the stirred bioreactor using a 250 milliliter volume glass reactor. Use a digital peristaltic pump or similar pump with an 8-channel pump head to control medium exchange. Assemble the reservoir and modified bioreactor lids from hard and autoclavable plastic with stainless steel pipe pass-through ports to